All right, hello everyone, and welcome to this session. I'm going to briefly speak to you about Omics Logic and our training that is available that is hopefully going to be useful to some of you that are joining today's session and in general. So I'm going to get started. I know that we maybe have another minute or two, and welcome for others to join as well. But let me briefly introduce myself. My name is Ilya Brodsky, and I am the CEO of Pine Biotech, one of the co-founders of Pine Biotech, and have been doing a lot of different types of activities around bioinformatics research support, development of the T-Bioinfo platform in application to different challenges in bioinformatics, and a big and growing kind of area of our work is related to training and education. And so today I'll talk specifically about the training and bioinformatics and data science education. And I'm also looking forward to getting to know a little bit more about you and who you are. Now, in addition to doing this for Pine Biotech, I'm also an instructor at the Georgetown University School of Medicine, where I teach a course on machine learning and Python. And I also work together with the Louisiana Biomedical Research Network and the National Association of Idea Principal Investigators to bring this kind of training online virtually across many different universities in the network to help elevate the level of biomedical research across these universities. So here are some topics that I would like to cover today. First of all, I'd love to talk about the role of data in the research lifecycle. So really, how does that address the issue of time and efficiency? Time is the most valuable asset that each one of us has. And when you think about time in perspective of a project, in perspective of your career, and in the perspective of getting to complete a certain valuable asset, for example, a publication or a poster presentation, or maybe a grant, these are very real considerations about time and efficiency. The second topic I'd love to talk about is to be or not to be. Should I become an expert or a generalist? So I'll offer kind of my perspective on this, what I have seen across thousands of students that we have dealt with as we run the Omics Logic training. And then I'll talk a little bit about the psychology of getting started. So I think that, if we really look at the issue of why is it so difficult for people who have not had the experience from an early age dealing with data, it becomes difficult because a big part of it is the fear of solving irrelevant problems. And I'll talk a little bit about that in the context of other available types of courses that are out there. Then I'll talk about the future of your and my academic careers. So what are the realistic prospects for someone who has an expertise, perhaps a deep expertise in a particular area of research to really succeed in the academic career? And if you don't succeed, what is plan B? How do you plan for that? And how can you prepare for something that can be equally as rewarding and impactful? And then finally, I'll talk about courses, lectures, and training. Are they really enough? Is it really enough to have a certificate to show that you have completed some training? And does that mean that you have the skill? And what does that tell the potential employer, the potential collaborator that you will have about your expertise and experience? This is the last point. The last point is about a community. And the community is really all about exchange, giving and taking. So I'll talk a little bit about how we are trying to grow Omics Logic into more than just a platform for learning. We're also looking forward to working with collaborators and making sure that this community grows organically beyond just what our company can provide. So with that, these are the topics for today. I'll kind of refer back to the slide a couple of times throughout the presentation. And I hope that if you have any questions along the line, along the way, if you have any comments, any questions, feel free to add them in the chat. I will ask at some point for you to do some practical things and maybe refer back to the chat box. So please keep that open and keep that in mind. All right, so again, thank you, and let us start talking about the first point. So the role of data in research lifecycle. So I think traditionally we think about two separate areas that are data generating and interpreting or making decisions about. And I think that this kind of a separation has really kind of brought us to the point where we are today. So today, we really see that all of these breakthroughs in techniques and new technologies in molecular biology, precision medicine, biotechnology, and agriculture are causing for there to be a gap between data and information. So what do we know how to generate, and what do we know how to interpret? And in fact, this is not just based on my perception. This is something that has been extensively published on, including this interesting survey that talks about how technical demands are actually preventing learners from acquiring these relevant data analysis and data interpretation skills. So it starts from this kind of conceptual separation of the two parts where we generate data and who analyzes and makes decisions about the data. And then we'll talk about the specific skills that are needed, because obviously to generate data, you need to understand the wet lab. You need to understand how the samples are prepared. What is the experimental design? What are kind of the biological objectives that you have in mind? And then when it comes to data analysis, it's a whole another world of algorithms and data types and different types of data visualization techniques and everything that goes into interpretation. So it's really another world. And because of that, we have this gap that actually causes people to think, well, you know, if I want to get started with data analysis, with bioinformatics, I have a whole list of prerequisite courses that I have to complete, like mathematics and statistics and coding. And then I have to have someone to guide me and to help me. And there's maybe limited infrastructure on where to store this data and how to access it. And also, how do I work on data sets that are relevant to my area of research and not just you know, some hello world or marketing or finance data that might not be relevant to what I'm interested in, and I might find kind of boring or even difficult to kind of try and understand the concepts that they're working with. So to summarize, I think that, you know, there are specific challenges and limitations when it comes to introducing this kind of data science curriculum for life science students. And I think that it has to do with incredibly dynamic field that bioinformatics is, with a lot of new techniques and a lot of new software that is being developed and updated all the time. I think that, you know, typically universities and academic institutions are limited with their processing power and how much of that power could be allocated to students. There are not a lot of curated data sets and effective pipelines that somebody has validated. So you kind of anticipated a lot of technical challenges when you start analyzing data, and you don't have examples that are maybe relevant specifically to your field. I think that students, even if they are introduced to kind of theory of bioinformatics and data science, most of it is theoretical, and they don't have a lot of practical experience because they are placed in projects to perform very short-term and specific tasks. So they don't have that broad view of what goes into all of the steps combined. And then I think that, you know, faculty, because they have experienced academic growth, you know, maybe in the previous few decades themselves, they don't see the need specific to the students' challenges that they face. They don't really kind of appreciate how effective it could be if students were exposed to these specific types of skills that are needed, and are not kind of systematically thinking about how to address that challenge. So what we are trying to do is to address some of these challenges through these three elements: standardization, organization, and systematization. So what does that really mean? I hope that in the next few slides, I'll be able to kind of expand on all of these, but in a nutshell, it means that the curriculum needs to be standardized, and it needs to be kind of the same thing, whether you are studying neuroscience or you're studying agriculture, two very different fields, but both of them require the data analysis skill sets and tools to be able to make decisions about the data that they generate. Organization. I think that there are a lot of online tutorials, little snippets of code here and there, some publications about pipelines, but no one's really put the effort to organize this in a way that anyone can go from a very beginner level and kind of advance to a more expert level in a way that is predictable, kind of clear, and has validated points of progression. And then finally, systematization. So systematization means that now this package can actually be turned into a system. So it's a package of online tutorials, a package of pipelines that you can run, code and exercises that you can complete, and projects that you can develop. And that system is really what OmicsLogic is all about. So we talked briefly about the role of data in the research lifecycle, about time and efficiency. We talked about the gap between those that generate data and those that analyze data. We talked about to be or not to be. Should I become an expert or be a generalist? I think the answer clearly is that you should be both. You should be an expert in one field, but you should also have the general understanding of trends in this important field where we are. And finally, let's talk about the psychology of getting started, overcoming the fear of solving irrelevant problems. So what is that fear? Probably you have tried at some point in your career or life something new. And it always starts with this mock example that has nothing to do with what you are interested in. And so that's what I want to talk about how you can kind of flip that and start from where you already are, maybe in a field that you're interested in, and then from there identify the gaps that prevent you from acquiring the skills that you need to be not only kind of superficially aware of the challenges, but also being able to solve and overcome those challenges. So then after that, we'll talk about the future academic career. What are some limitations, maybe challenges, and some opportunities for those that do have the skills and can validate that they have the experience or expertise, and about how we can kind of view coursework as just a starting point, and then what else is needed to be enough, to have enough experience and to know that you can apply these skills, not just, kind of know about them. And then finally, I'll finish with our community. What is this community? Who's a member of this community? How can you become a member of this community, either as a faculty or as a student? So let's talk about the psychology of getting started. How to overcome the fear of solving irrelevant problems. So according to the Bloom's taxonomy of learning, learning is a process that has very specific kind of layers that go from just the basics, understanding terminology, remembering terms, and kind of that's where it starts. So for example, in my own experience, when I started dealing with interacting with a lot of biologists, I remember quite a number of conversations where it seemed like I have to have a dictionary to interpret what they're saying. It was about genes, it was about proteins. I remember I had a conversation with a clinician that was talking about different cancer immunotherapies, and literally every word in a sentence I needed to interpret. And so I think that, even if you don't understand all of the terms, you need to be familiar with them to be able to string together what is the actual meaning of this sentence or of this topic. And from there, of course, you need to start explaining ideas and concepts. So we'll talk a little bit about how the same applies to data. When a biologist speaks to a data scientist, for example, I think the situation is very similar. You kind of listen, and all of it is about different technical details and terms that maybe they're so familiar with that they feel like everybody should understand, but you feel like you actually don't know anything what they're talking about. And so that's really the foundational layers when you start to kind of recognize and understand some of these terms and the meaning of them becomes clear. Then you can use this information in new situations, right? So next time when you go and you read an article, a publication on the topic that you're interested in, instead of skipping the methods section, you actually read through and you recognize those same terms that you just learned, and you understand how they are applied in that particular context. So then you all of a sudden start seeing that everybody's using these terms. Everybody's talking about the same kind of processes, about data cleaning, about processing, about kind of visualizing this data, about statistical methods to confirm their hypothesis, and things like that. And then finally, you kind of go on to the peak of this pyramid, which makes you independent. And that independence and that ability to apply what you have learned to completely new situations leads you to the ability to produce new or original work. So with this in mind, let us get started. So now I would like you to all take that leap of faith and come with me to the Omics Logic portal, where you will find all that you need to get started with this process. So in the chat box, thank you, Ajasvi, for sharing that link. There's a link to the portal, which is called Omics Logic Biology as a Data Science. So to get started, all you need to do is to sign up. So when you get to this link, you click on Login, and you can create an account using your social media accounts or email. So I'll give just a couple of minutes for anyone who would like to sign up right now. I will sign in the meantime, so that we can go through, and we'll use a couple of practical exercises from this portal to give you a sense of what is this all about and how does it work. So please take a moment to sign up. After you sign up, you will be redirected to the courses page. And when you are on the courses page, you should be able to see your email address or your name right here. And so the next thing that we're going to do is we're going to go to that profile page. So again, I'm going to give this just a couple more minutes for anyone who would like to sign up today. And once you do sign up, please let me know. You can simply put done or yes or number one or something like that in the chat box, so that I know that you have signed up and you're ready to go on. All right, thank you, Mrinal, Saja. Yes, and if you already have a profile, then you're good to go, but we're just going to give it one more minute for anyone else who would like to. All right, great. So thank you. So when you arrive on this page, what you will see is that you have a profile that you can complete. So as you can see, my profile is 100% complete. So the first thing to do, I think, is to call this place your own. Okay, so if you have an issue signing up, Maybe you can share your email address. Our team is here in the chat and they can maybe help you with that as well. So, okay, thank you. So once you sign up and you complete your profile, it helps you kind of connect what you are doing to a profile that you can share. And we'll go through a few of these, but I'll kind of show you around what goes into completing your profile. Obviously, your full name, accurately capitalized, some kind of a picture. It doesn't have to be your profile. It can also be a logo from your university. Social media accounts or org ID, where you can connect to your academic publications. And the way you edit all of these is by clicking on this profile button. And here you can see all of this information that you can update and edit. Now, as you go through the different exercises that we have, you will see that there are points, achievements, and certificates that you are able to achieve. You'll have the option to get a subscription. So today, anyone for free can do the exercises that we are going to do. But if you would like to go beyond that, there is a basic subscription, just at $10 a month. And then you will also see how you can participate in programs where a mentor will guide you through more and more advanced topics that you might want to know how to learn. So all of that information is going to be also in these tabs. Here you have courses, programs that you are a part of, certificates that you have obtained. And once you have a certain number of points, you will also be able to develop a project of your own, which I will get to in just a couple more minutes. So this portal allows you to access our community. Beyond just the portal itself, we have a LinkedIn group with over 2,000 professionals in the field of bioinformatics and machine learning and data science, a community of about 10,000 people on Facebook, and you will also join kind of our community on Slack, where we discuss different topics related to this field. So as I mentioned, you want to start by completing your profile. That's where you will see your activity and validate your achievement in the form of certificates. And the next place that you want to go to is the course tab. So under the courses, you will be able to look up specific topics that are most interesting and relevant to you. So here you will find a search bar where you can just search with any text. You will also find some topics that are kind of used as tags for the different resources that we have. On the T-Bioinfo platform, transcriptomics, project examples, bioinformatics, and others. And down below, you will have collections. So the first collection that I want to introduce you to today is called this Getting Started collection. So it's right here, Getting Started, and that's what that logo looks like. The Getting Started collection is something that we developed as a free resource for anyone who would like to jump into this field of bioinformatics. And it starts off with a very brief course called Introduction to Bioinformatics. The Introduction to Bioinformatics course covers the very basic ideas about how bioinformatics has impacted fields like precision medicine, space study, and others, and gives you a view of the challenges that big data in this field introduce. The next course is called Bytes and Molecules. Again, it's a free course that you can complete and get a certificate for if you just signed up. And this course offers a view of the basic terminology. What are genes and what are bytes? What are data concepts that you need to know? How do these things fit together? And kind of what are the specific concepts that you need to understand? It also gives you a view of how different coding languages like R and Python are used to actually work with this data more effectively. It's not like you cannot, look at sequences of genes in a notepad or write them out or print them out, right? The first genome is actually a collection of books that you can store on the library. But to be more effective with these kinds of data, it is very useful to be able to work with these object-oriented languages that also have a lot of useful packages that you probably know about that generate publication-ready visuals that you can use, and also produce easy-to-use statistical analyses and machine learning methods. So the Bytes and Molecules course covers some important aspects that I think anyone would benefit from reviewing and refreshing in their minds before you jump into the next series of courses. Now, what we typically recommend is after you complete that course is to go through some of the additional coursework that is aligned with your interest. So that means you look up not just skills like Python and R, but you actually focus on the types of applications or data sets that you might want to work with. For example, Ebola virus, cancer, or maybe neuroscience like Alzheimer's disease. So to summarize, the Omics Logic basic curriculum includes training on introduction to bioinformatics and data sets like genomics, transcriptomics, metagenomics, and epigenomics, and an additional set of training courses on the basics of data science, coding, statistical analysis, and machine learning in application to biomedical data. Now, the important part of it is that we prioritize this approach of code or no code bioinformatics. And I think that's very important because, as I mentioned, there's a psychological barrier that prevents many people to get started because they anticipate the difficulties of actually learning the basics of coding, math, or some kind of a portion of this that just prevents them from getting started. So we want to eliminate that altogether. And that's why we have a series of courses that you can complete on all of these topics without coding at all. And that helps you understand the logic of analysis and the application of these analyses to big data, which oftentimes is not even possible on your own computer. The second part is, well, what happens behind the scenes? And behind the scenes, you will see that each one of these steps is actually a series of algorithms or scripts that string together different analysis topics so that you can actually perform the analysis from beginning to the end. A large part of that, when you become more independent with these tools, is to be able to start asking your own questions of data. And that means a lot of data manipulation, data wrangling, visualization, statistical analysis, etc., that you have to know how to do. And that's why we provide an extensive training on data science in R and Python. And then finally, as I mentioned, we have a community of experts that teach, that share project examples, and that talk about the cutting edge analyses that are needed in the space where the challenges and opportunities are for anyone. And we turn that into a collection of resources that anyone can use once they are a part of this community. So let me briefly introduce you to how practically does this work. So the first example is going to be from the T-Bioinfo platform, where I will show you how you can play around with data. So what we'll look at is a series of visual outputs that you can play around with to understand what this data means. So how to go from analysis to interpretation. And then I will take you through a brief exercise where you will see how you can get started with coding, which means solving coding challenges, understanding code syntax, and being able to take that code and use it on your own computer or in your own cloud environment. To do that, let's go to the T-Bioinfo platform. Okay, which is a separate portal to the Omics Logic portal. But after you have signed up, you will get credentials to log in here. Thank you, Jessley. So you have the link to this portal in the chat. And as you kind of get started with bioinformatics, one of the objectives is to understand a whole variety of possible analytical approaches to different data types. So that's why this areas of analysis section is organized by data type. You can see here next generation sequencing data, mass spectrometry, structural biology, data integration and modeling, And here you will find some additional sections as well, like virology, single cell analysis, cancer biology, and metagenomics as well. So it's really a comprehensive platform that is organized, driven by logic of analysis that provides us with some practical tools that you can use for research, guided tutorials that you can follow, and also interactive dashboards that you can use to kind of see the relationship between the data and the biology that you are studying. So to do that, let us go to the transcriptomics section right here. We're going to go first to the bulk RNA-seq example. If you click on the drop-down, you will find several demo pipelines that are essentially projects that you will also find on the Omics Logic portal. And here what we can do is we can take this second project called PDX. This is a project on breast cancer, and I'll just use it as an illustration of how this platform works in application to teaching, and then what kind of data exploration you can actually get as a result of performing this demo analysis. So as you will find, each step of the way includes logical explanations of the steps without being focused necessarily on the technical terms. For example, in each one of these steps, pre-processing, mapping, and quantification, a number of alternative algorithms can be used. All of these alternative algorithms perform the same function: pre-processing, mapping, or quantification. And so you will see that each one of these steps is color-coded to reflect the function of that algorithm. When you click on OK, you will then be given a limited number of suggestions. So you don't have to search around and think, how do I connect this step to the next? It automatically suggests what is the best next step for you to do. So here you have an explanation of this particular step, which is to clean up the reads and make sure that the read quality before you start processing it downstream is good. Here you have an explanation of why these technical artifacts might arise. Then we will click to mapping on transcripts, where we have to map reads to a particular region on the genome. And then we will quantify how high of an expression do we have for a particular gene. So for example, if I want to compare two conditions, I would like to arrive at differential expression. So here we have the differential expression, where we will look at some statistical measures that can tell us which gene is actually responsible for the differentiation between two different conditions. And then finally, we will click on End. And here we will look at some outputs that you can go through and quickly recognize probably, so that you understand what is, for example, the output of Trim-O-Matic. How do we evaluate the sequence quality of these reads, and what is actually being trimmed. So here you can see the multi-QC report. And then you can also check, for example, the level of expression. How does that expression vary by the different samples that we have here? So you can see that fairly comparable to each other, so we can proceed. And then finally, let's go to this link, which is a dashboard. Now, this dashboard combines together several of the analysis results so that we can start looking at the connection between the data and the interpretation that we are trying to make. So here we can, change different parameters that we have. We can adjust things, and we can kind of explore the different aspects of this data set to really try to link it to the biology that we know and understand. Okay, so here we changed the color scheme, and here we also see on the volcano plot how individual genes that are responsible for the difference between these two conditions are annotated. So we have the original gene ID, we have the gene name, and we also have the common name, so we understand what this gene does. Now, of course, this is useful, right? So for example, here I can add a couple of other genes and kind of see what they mean, and all of a sudden I recognize, oh, there's a gene here that I know something about, EGFR, and EGFR seems to be highly expressed in group 2 and not expressed in group 1. And so I can start making interpretations about what I see in this data. Now, this is nice because I can play around with things, but it's also good to have this data on hand. And so here you can download all of this data together. Here's another project like this. This is a project about the differential gene expression results for an Alzheimer's study, where we have 2 conditions, and we want to identify the top differentially expressed genes between these 2 conditions. So people who do not have Alzheimer's disease and people that do have Alzheimer's disease. And we have a clear summary here of how these particular patients vary. And you can also see here the different genes that might be responsible for that. And you can kind of compare 2 different genes, for example, that are up and down regulated in these different groups. Another example that I have here is a metagenomics data analysis. So again, a different type of data, not RNA-seq, but describing the microbial community. But what I can see in this microbial community, if I look over here, is that there are different ways that I can actually analyze this kind of data. There are many different types of metadata annotations for each one of my samples, and there are many different types of metrics that I can use to compare and contrast the different groups in my data sets. So this is just a preview of how very quickly you can start looking at data, using annotation, understanding the major differences between the data sets that you are analyzing, and kind of get familiar with what might be a standard visualization that you will find in publications. Let's take a look now at the second example. So let's say we understand conceptually what is being done. How can we use that conceptual understanding to actually start learning how to code and analyze this data on our own? So to do that, we will go back to the Omics Logic portal, and let us go to the Courses tab. On the Courses tab, we will start with Introduction to Bioinformatics in R, getting started with bioinformatics. So getting started with bioinformatics is a course that you can complete independently in probably under a couple of hours. But what it gives you is a preview of how you go from kind of looking at charts like this to understanding the concepts behind them, and eventually being able to practice them independently. So we just saw the result of this pipeline that basically showed us the RNA-seq gene expression profiles across several different samples, right? So we saw something like this, where we have a lot of samples and they have a distribution, and we can use that distribution to compare them to each other. And so one of the plots that we were just looking at is a histogram, or a bar plot, for example. And so in this example, you can actually start practicing this code on your own. The console is built right into the lesson. So all you have to do is follow the examples and replicate the syntax that was given to you. So here, for example, you already have a full script that you can simply run. But you can see that there are portions of this code that are missing. And so the challenges that you will see is how to complete this syntax in a way that, it actually works. And so to do this, we need to write in read. So here I'm loading the data by reading this table that is on this link. And you can see that it has row names and a header. And so here, I will add true. Okay, and now I get another issue, another problem, and you can see that true is actually not spelled correctly. So what I need to do is I need to make it true like this. So I'm learning the syntax as I am trying to run this analysis. So let's clear that output. Okay, now you can see there's again a problem. So there's something missing here. There's not an X argument. So here I need to say is matrix df. And finally, I am successful, right? So the process of learning actually happens through troubleshooting the errors that I am getting, right? And so what I'm learning about this is how the code is organized. How can I just try and run it and see what happens? And when it happens, I actually see the problem. And once I see the problem, I can address the coding issue that I am facing. So in a similar way, you can now address the second problem that we have. Again, you will find all the same kind of ideas where you have different kind of missing pieces that you would have to fill in. As a result, you can kind of read through the errors and try to see whether you can find what is the error and what exactly is the problem that I am facing. So here again, the same idea. You kind of go through each one of these steps, complete them, and after you are done, you can actually see your progress in your profile. Now, in addition to that, you can see everybody's progress, and you will see that this is not just a platform that we use, you and I, but you can see here how maybe every minute there's someone who is doing some kind of an assignment, so completing some kind of a course, signing up, completing quizzes and other types of activities that we are just looking at together. So really, this is just a way to get started and join a full community of people that are on the same path to learning about data science and bioinformatics. So as you will see, a lot of different options to start playing with data and kind of trying to think about what it means, and options to start playing with code, where you can start looking at how to fix syntax, how to run a particular script, and even how to take that script and use it further in your own research by knowing how to load data, how to visualize data, how to perform statistical analysis, how to do data wrangling, and eventually also how to perform complex machine learning algorithms and apply those models to your own data sets. So it's great, right? But is this kind of an approach enough? So this is a picture of me a few years ago when I received a certificate for completing an online course. And today I have maybe tens of these certificates of courses that I completed on data science and business and finance and all these other topics. Does that make me an expert? So indeed, there are lectures that we go and see, YouTube videos, online tutorials, Coursera, edX, Udemy, and continuous education. But do these show that we know what we are doing? And do they actually give some validity to the skills that we say that we have? My conclusion from going through these courses myself and working with many others that have completed this kind of training also is that so many times it is not enough to show that you understand a topic or have that skill. And that's why I think the portfolio approach here makes a lot more sense. GitHub, Kaggle, LinkedIn, research posters at conferences, publications, those are things that actually add validity to us knowing how to do something. What can you see in a project? Well, in a project, you can actually see that you've understood the problem, or you have yourself kind of created a question about a particular problem. You have been able to overcome challenges, troubleshoot, revise, understand how this process of analysis works, right? So you've put in the effort. And then finally, it's your presentation as well. Either it is written presentation, visual presentation, or live presentation like I'm doing today. You have to validate that you can showcase, explain, answer questions, and be able to support your statements with evidence. So those are the things that actually help us gain that experience, but also help us show that we possess the skills that we say we have. And that is why this Omics Logic portal is not just about training skills. It also has project examples and curated data sets in the field of oncology, infectious diseases, neuroscience, and agriculture. And these topics are actually the beginning of taking that initial kind of training to the next level. And that next level is creating an independent project based on a topic that you understand, you care about, and you know how to present to others. To do that, on your profile, after you complete your profile, right, and we just kind of saw how completing the profile happens, you have to upload your image, you have to add your bio, you have to connect your social media links, right? All of these steps help you create a profile. And after you create that profile, you can see that the next step is to gain 10,000 points. So to gain 10,000 points, you will have to go through courses and training to actually perform the exercises. And then finally, there's a course that will introduce you to the basic idea of how to actually do a project in bioinformatics. And after you've completed those steps, you will be able to start working on an independent project, which is like preparing for a publication. So you actually take a project title, start kind of looking at specific data sets and maybe doing literature review and going through steps of actually preparing a full kind of an overview with a presentation of your analysis results that you can then add into this project of your own. Now, it includes addressing some very important challenges in this field of bioinformatics, like, for example, reproducibility, being able to provide, exactly which data set did you actually use for this study? Or did you curate the data in any way and maybe took just a portion of the data or cleaned it up in a certain way, right? So all of these things are covered in that section. Now, instead of just listening to me talk about it, I would like to share with you some of the projects that some of the people on our portal have completed. And then, Mohammed, I will actually get to your question there in the chat as well. So here's a link where you can see all the different projects that students who have gone through this training have completed. You can look at this project and kind of see all of the different steps that we just described. So where are the data sets that you can actually download, an abstract, an introduction, a methodology, what was done in this project, which pipelines they have used, what do these results look like, right? So similar to kind of what we saw in those dashboards, etc. And you can really go through all of these steps, and importantly, you can react to them. You can support the person who did that project by liking, commenting, or even sharing that project with others on social media. So again, take a look at some of these projects. You will find that these are all students who came from a life science background, very quickly learned about these new topics like processing, analysis, and visualization of this data, and were able in a short period of time to create a project and present it to mentors for feedback. Now, when I say mentors, what I mean is we have an independent process where you can do this completely on your own, just following the steps that I described. Or if you would like to do it faster, you can work with our mentors to actually help you select the topic, address technical challenges, and review and provide you with feedback on the final product. And that is where programs come in. So if you go into the programs tab, you will see here mentor-guided programs on different topics, including what we call a research fellowship. So the research fellowship is really a 3 or 6 months process where you work with an expert mentor who has a bioinformatics or a data science background and can introduce you to additional mentors on the topic of your choice, like oncology, neuroscience, biotechnology, and others. And so the process starts by a little bit of training, depending on what level of training you already have, introduction to the different types of data sets that are out there, and then eventually being able to work through those data sets to create a project of your own. So let me also give you a brief overview of some of these mentor-guided programs so that you understand how these mentor-guided programs are different from asynchronous independent courses that we just looked at. And then I will finish off with how these programs fit in with the research fellowship and the community of experts that you can leverage to prepare your portfolio of projects, either to advance your academic career or explore careers in the industry. One of the challenges for many people coming from a life science background is to understand that a lot of these analyses divide into specific challenges of that particular data type and general skills that are needed across the board. And so a good example is transcriptomics, where we have just what we saw, the standard approach to analysis, kind of standard statistical analysis, and machine learning of complex RNA-seq or gene expression data. And in this program, we cover all of these sections by going through the logic of the analysis. And then after we understand the logic of the analysis, understanding how this varies, whether we analyze bulk or single-cell RNA-seq data. We understand demo pipelines, where we kind of string together individual steps into a logical flow. And each one of those steps has a specific role that allows you to understand how these steps are connected to produce a result that you can interpret. And then you're introduced to projects where you can focus on analysis of such RNA-seq data in cancer, in neuroscience, infectious diseases, or even agriculture. Now, as you all know, the single-cell data is becoming more and more available. And so the question is, how do you actually take this kind of data, analyze it, understanding all of the steps? And so we have created pipelines that kind of organize all of this information in a simple to use way so that it is reproducible and clear what the logic of the analysis was. And then, as we already saw, you have these dashboards that come alongside the standard scripts that are going to be used in R and Python to perform this analysis independently, but also to be able to apply standard tools to different data sets for data visualization and your presentations. And so you will have access to interactive notebooks to kind of complete that on your own. Finally, you will be introduced to project examples. These project examples illustrate how these challenges have been addressed by experts on what types of data sets and leading to what kind of interpretable results. So these types of programs are data specific. Another set of programs are application specific, like bioinformatics for infectious diseases, where we actually take application of bioinformatics to different types of infectious diseases that are either viral or bacterial in nature, to understand the process of pathogenesis and also the interaction between the host and the pathogen. So there we take examples from different types of diseases like malaria, tuberculosis, COVID, and others. And we learn about these approaches that are commonly used for diagnostic purposes, as well as to understand the basic biology and design interventions. So here we have a set of projects that we have prepared together with our collaborators, like the enterovirus outbreak, EVD68, malaria infection in Ghana, SARS-CoV-2 genomics around the world, and the Ebola virus pandemic throughout the West African outbreak not so long ago. So what's important here is that you are going to understand the basic approaches, but you will also be introduced to specific types of analyses that can be used for practical applications. For example, generating a variant report. So we take a report from a company, Cosmos ID, and we replicate all of the steps of this report in our studio to have the exact same output and all of the code associated with it. But we also go beyond the sequence analysis, where we start going from sequence to structure. How do we actually superimpose this variation at the sequence level to be able to understand the value of having a protein structure that helps us interpret the interaction between the host and the pathogen, or the immune response that we observe in the body? Now, finally, we have a comprehensive set of resources for training in data science. And this is actually a course that I also teach at Georgetown University using these resources. And I find them extremely valuable for the students that want to go and work in the medical field, biomedical research, and biotech companies. So I'll talk about that in just a few more minutes. We already saw getting started with R and Python, where you can get started with code and learn how this code applies to different types of data. We saw RNA-seq data, or we can look at sequencing data. The code itself is explained in chunks. Then you are given exercises to perform visualization. And you also see the application of this approach to different types of challenges: metagenomics, transcriptomics, and others. So this course can be taken in R or in Python, and it covers these standard topics like data wrangling, machine learning, supervised and unsupervised learning, feature selection, signature patterns, regression, deep learning, and others. And kind of separate from the other types of courses that we've had, this one is also focused on going through these practical coding challenges, and generating a number of notebooks that you can use as your own repository of projects that you have completed. And these projects are something that you can run and kind of know the analysis yourself, but also anyone else can try on their own. So this is, these are just some topics that we cover in this program that I think are very relevant as we have more and more complex data sets that you have to analyze. And that goes from kind of a high level overview to more detailed kind of understanding of methods that I think are important for anyone who is going into this field. So it's not just high level superficial information, but actually detailed connection between the methods, the way to use those methods, and project examples where you can apply these methods in a meaningful way. And so the way that these projects are designed is that we take all of these concepts that I just went through, and we actually use specific publications that reference these key terms. Like here you can see an abstract from a paper that we actually use in the exercises. And these papers lead to an understanding of how these methods are being used by experts. Let me finally summarize this with who and how these programs are organized and what are the practical outcomes that I see. So typically, a program consists of 3 steps. First, we understand the context. Then we learn about bioinformatics methods. And then you have a series of exemplar projects that you can choose from so that you can get started on a project of your own. And so the full process is when we start from basic training to then get into more and more expert kind of areas, which we have guest lecturers that are going to speak about those topics. And then you are prepared to work on a project of your own that results in a showcase. So we've been doing these programs with a number of organizations and universities around the world. Here in Louisiana, we are working with the Louisiana State University and the Louisiana Biomedical Research Network. We've worked with, and we continue to work with, Georgetown University Medical School, and we work with international universities like Amity University in India, one of the largest private university systems in India. And because we work with a lot of these collaborators and academic partners, we have now developed a network of experts that regularly contribute to expanding resources and topics that we cover. We have a biannual event that we call the Omics Research Symposium, where members of University of Haifa have presented as well. And they come from esteemed other kinds of places to really talk about the cutting edge. Where is this whole field going, and how is it being used in a variety of fields? Now, I promised to tell you a little bit about alternatives to the academic pathway. I think for academic pathways, we've worked with many students who have applied bioinformatics to research problems. But sometimes that career can have challenges. You might not find the right mentor. You might not be able to do a postdoc multiple times. And most likely you will not get tenure, at least for a lot of people that does not happen. So what is the alternative? Well, I think the alternative is to focus on transferable skills that are needed in research, but also valued in the industry. And so we have recently started looking into the specific industry-related skills and projects that would help our students kind of develop a portfolio of projects and find a job that they can feel that they can contribute to. So on our portal, you will also find a listing of jobs, starting from internship jobs all the way to more advanced industry jobs for more senior professionals. And finally, I want to tell you about an upcoming event that our team has organized, the Microbiome in Health and Wellness Hackathon. This is especially relevant to those who are innovators, those that want to leverage the techniques and technologies that we talked about today from computer science and biotechnology, but add a portion of business to it as well. So the hackathon, and Ojasvi, if you could share the link to the hackathon as well, is going to be focused on these three topics. So it's preclinical microbiome, in obesity and nutrition, skin aging and beauty, and pregnancy and femtech. The way this will work is that a team of at least 3 students should participate to take on one of these challenges and develop a minimal viable product. So you will see all of the details about the microbiome hackathon on that link, and we welcome you to register. We have some guest lectures that you will be exposed to every aspect of this process, and the upcoming lecture is coming up. So here below, you can see all of the lectures, and I think the upcoming lecture is actually later today. So Jasvi, maybe you could share the details on how to register to that upcoming lecture. I think the way that you would do that is by filling out this form below to get more information about this upcoming lecture. So I know that I've taken a little bit more time than I wanted to, but let me go back to my original slide and finish off with this. So today we talked about the role of data in the research lifecycle, time and efficiency. There's more data, less people to analyze this data, and the way to maximize efficiency is to have people that can handle both. To be or not to be, should I become an expert or be a generalist? I think the answer is that you have to be an expert in something, but you also have to be a generalist when it comes to data. You have to understand many different types of challenges, many different types of approaches to those challenges, and many different data types. The psychology of getting started. I hope that I alleviated the fear of solving irrelevant problems by showing you how you can get started by doing all of the things that you need, understanding the logic of analysis, learning how to code, and applying those skills to a project that you care about. Then we talked about academic careers. Is there a future, or are there some alternatives? And so we are going to, you will see how there are opportunities both in academia and in industry, and having those transferable skills is key. Our courses, lectures, and training, not enough. So we talked about the portfolio approach, how instead of showing certificates and courses that you have completed, you focus on completing projects that show what you know and how you did it. And then finally, we talked about our growing community, where you can both give and take. And so with that, I want to welcome all of you to register, participate in all of the events in the online portal that I just demonstrated, but also to contribute. So if you would like to be a trainer, you would like to contribute a project example, or work with us to develop better solutions to address these challenges, I welcome you to contact us. So I'm going to share my email address. All right, thank you, Sparsh. And anyone else who would like to connect with our team, we also have a marketing team. Some of them are here. Sparsh, Dar, Ojasvi, Duta, and Ayushi are all here. So our contact information is here, and we're looking forward to interacting with you further after today's session to learn more about your background and your interest in what we presented today. So let me stop the sharing. And if anyone has any questions, I will stay here for the next few minutes and try to answer those. And I don't know if Julia, you would like to add anything to what I just described. You're on mute. You're on mute, yeah. Oh, let me hold on 1 second. Yeah.
Thank you. Hi, Ilya. Thank you very much for your talk. Unfortunately, for for everybody joining from Israel, there was a bit of a confusion. We had a change of time, so maybe some of the people joining from Israel just joined now, but hopefully this session was recorded, so maybe you can also share a link where it was recorded so that you know, whoever missed it, like myself, could watch it again and see all the great information out there. So thank you.
All right, great. Yeah, sorry about the confusion. So here's the link. The link is in the chat. Thank you, Ojasvi, for sharing that. And if you do have any questions, you have our contact information here in the chat as well. And we're happy to talk to you more about any of these topics that we covered today. So thank you, everyone, and have a good rest of your day.
Thank you very much.